Hi, welcome to Module 2 of the Research Data Management for Microscopy training resource. By now, you understand how to create meaningful file names for your microscopy data. But where will you store and how will you organize these files? In Module 2, I'm going to talk about directory structures. A directory structure is an organization of your files into a hierarchy of folders. In a well-designed directory, there's going to be a logical sequence of folders, and hopefully each folder is given a singular purpose. This helps to avoid redundancies and, importantly, the duplication of your microscopy data. Ultimately, a well-planned directory structure will maximize your research efficiency, as data is easy to file and hence easy to find, and you minimize the risk of using the wrong image version or the wrong data. So there are a couple of things you need to consider. One is how you can organize folders on the microscope, in other words, when you generate your data, and two, how you can organize folders thereafter. For example, in a lab or research project repository. Regarding one and your visit to Adelaide Microscopy, the organization of folders at the microscope often is determined by the microscope manufacturer. For example, whether the saving of data is limited to year, month, day folders. And so just to be aware, you may need to work initially within that framework. In general, each Adelaide Microscopy system uses the below directory structure. All data is written to a local drive, typically the D drive, but maybe it is labeled with some other character, and under which your microscopist will then prompt you to create a user folder. Now we recommend to name the folder in the format given name, surname. So for myself, that folder would be called Bryant Roberts. Now, within your user folder, I strongly recommend, though it's not essential, to create a folder titled Raw Data. Then, either within the Raw Data or user folder, you may generate subfolders per your own decisions or decisions of the microscope manufacturer, where you'll ultimately save the image data. Next, let's put the microscopy data in the context of a broader research project. So the microscopy data, that which you generate at the microscope, will not remain in the local drive for too long. Rather, you'll transfer it to your lab or your project repository, and within the repository, it may live among many hundreds or thousands of digital files in many different other formats that you create over the life cycle of a research project. So that includes files for admin, method development, data, statistical analysis, and for reporting. And it's typically from here where you access your raw data for further processing and analyses. So it's going to be beneficial to well organize these folders. In your directory and repository, I recommend that for each project, each microscope that you use, to save image files in a folder titled Raw Data. And it is here where you'll save the image data as it was generated from the microscope. And you should consider that data set immutable. In other words, it cannot be modified, and any subsequent image processing occurs on a copy of those files. And this just helps to comply with funder or institutional data policies, particularly when it comes to longer term storage of at least the raw data set. In the remainder of this video, I'm going to discuss options for organizing folders and the subfolders within that raw data directory. So option one is to save files directly within the raw data folder. Keep in mind that deep directories, that is, the nesting of subfolder under subfolder under subfolder, may not be optimal for you, particularly if you create very few image files, say less than 10 or 20 in a single experiment. In that case, it's fine if you keep the directory shallow and store images directly in the raw data folder. If, however, you create many hundreds of images, then option two might be for you. And that is saving images in subfolders as determined by the most important attribute. So in module one, you identified and sorted the most important attributes for your data, and we saw that it was beneficial for default sorting your files. Subsequently, we create subfolders per the attribute values and save image data in each folder accordingly. So I show examples listed and including subfolders created first per acquisition date, so in year, month, days, per location, so sorted by climate, that's polar, temperate, and tropical, and by the treatment or intervention type, whether it's images from specimens treated with drug A, drug B, or placebo. So option two may be better suited to your data set, and you'll do likewise for the most important attribute in your study. Now finally, also where you may have very many images is option three, and that's to save images in subfolders nested within subfolders that are organized by the two most important attributes. So consider in the example case if time and treatment are the two most important attributes for my data. If I know time is more important than treatment, then treatment folders, in my case drug A, drug B, are nested within each time subfolder. Whereas if treatment were more important than time folders, date 1, date 2, are nested within each treatment subfolder, and likewise. But now it's your turn. Consider any of the above 3 options and work out which is going to be best for your microscopy data. And I'm going to see you in our third and final, but possibly most important module, where I'll address the documentation of your microscopy data.